Western countries. So you know that even a bottle of water in India, what it costs and what it costs in Europe and US. So there is huge price difference everywhere. Similarly, to execute clinical trials in Western countries is much expensive compared to country like India. So that's why these companies are moving to India to set up their offices. Fourth is large and diverse patient pool and treatment population for clinical trials. Because as a country we are highest in a lot of diseases like diabetes, hypertension because of the population. So it's easy to get the trial participants here. Then the availability of medical pharmacy and science graduates in our country is highest. All every year the graduating students are much higher than any other country. So earlier, what was hospital 2 decades back is not now. Now many hospitals are having infrastructure as good as what the Western countries have, even the facilities. So that all these things are attracting this industry to come to India and grow. So who all can apply for clinical research? It is a specialized knowledge what has to be gathered after you complete graduation. So any science graduate is eligible to pursue this course of clinical research. But as a pharmacy graduates, you have an extra edge over others because in your graduation you have already learned 50% to 60% of this field already. So that gives you an extra edge over other health science graduates. So after pursuing clinical research, which are the areas which you can look for as a career? So these are the subcategories, so many subcategories which you can choose depending on your interest. It could be either bioequivalence centers where they do healthy volunteer studies, or you can choose pharmaceutical companies, clinical research organizations, various site management organizations. If you like to work 9 to 5 only on weekdays, Monday to Friday, then you can choose IT companies where they do clinical data management and pharmacovigilance. There are fields like packaging and labeling of investigational drug, central laboratories, be investigator site staff, and you can also choose to be a training center. So these are the multiple subcategories where you can choose as per your interest. Okay, next please. So I have just enumerated few companies as per the categories. So let's say some of sponsor pharma companies. Then there are multiple pharma companies who are doing clinical trials. I could just name few, but there are lot. There are clinical research organizations doing clinical trials in India. There are multiple site management organizations doing trials. If you are interested in data management, then there are companies. If you are interested in pharmacovigilance, there are different companies. So, this is an industry which is growing with the CAGR of more than 20% every year, and this is the industry which is creating job opportunities for people like us and helping people getting 5-digit, 6-digit salaries in a short time, because even if the start time remains same in between the different industries, how fast we grow, that depends on the growth of that industry. So what I see in general is minimum 20% is the every year in this industry for those who are working here. and you can compare with your available options, what you might have thought already. Because see, till the time we are in college, we are very secure, we are in secure environment. But the moment you complete the college, you pass out, then we realize, okay, where we are standing. And that is the time when our family expect us to do something in life, right? So, better prepare for it well in advance. Search which are the fields you can work in, zero down on it, and then by the time you pass out, you start working on it. Okay? So if there is any question, you have it later, you can ask me. I'm around for some time, or there is a booth outside. I have people there who can answer your question. Okay? Thank you so much for giving me time. Yeah.
As a symbol of our heartfelt appreciation and deep respect, we would now like to present mementos to our speaker, who have graced this conference with their presence and wisdom. May I now request Dr. G. Arunachalam, Principal, PGP College of Pharmacy, to kindly come forward and to the honors for presenting the memento to Dr. Sunil Ramesh Chaudhary, Director, MedStar Institute of Clinical Research, Pune. We would like to invite Dr. P. Balan to honor our chairperson, Dr. G. Arunachalam, Principal, BGP College of Pharmacy. Thank you, sir. Your presence has indeed added immense value to our conference.
Our next session is going to be started. Our next speaker is Dr. R. Adivelan, Professor and Head, Department of Pharmacology, JSS College of Pharmacy, Ooty. He is going to deliver the topic Preclinical Perspectives in Drug Discovery and Development. For that, we would like to invite would like to invite Dr. M. Sendil Kumar, Professor, Department of Pharmacognosy, to introduce our chairperson, Dr. P. Suresh Kumar, Principal, Sri Shanmugha College of Pharmacy, to the gathering. Please, sir.
Good afternoon, one and all. It's a warm, immense pleasure to stand before you to introduce our chairperson, Dr. P. Suresh Kumar. Dr. P. Suresh Kumar, he was started his career in pharmacy from diploma. He was completed diploma in pharmacy in Glover College of Pharmacy in the year of 1998, followed by in the year of 2001, completed a B.Pharm in Periyar College of Pharmacy. In the year of 2006, completed a M.Pharm in pharmaceutical therapeutics in Annamalai University. Then followed by he was completed his PhD in the year of 2015 in Bristol University. Currently he is a principal of Shanmugha College of Pharmacy. Dr. Suresh Kumar, he was guided around 37 students M.Pharm and 17 PhD and 17 PharmD students and 1 PhD students he was guided. And he has around 60 publication papers in the national and international papers, and he was conducted in many conferences and seminars, and he was started his career in the year of 2000. He was started his career in pharmacy as a lecturer in 2007 in Rajasthan Pharmacy, Jaipur. Then followed by he was worked as a vice principal and HOD in Brown College of Pharmacy, Telangana, around 12 years. Then 1.5 years. He is also our alumni and family member of ECP, ERODE College of Pharmacy. Why? Because he was worked 1.5 years in ERODE College of Pharmacy as a professor in the year of 2001 to 2021 to 2023. Then he was joined as a professor in Shanmugha College of Pharmacy in the year of 2023. Currently, in the same college, he is promoted as a principal. And he is a membership in the various committee like APTA, IPGA and a member of Animal Ethical Committee. With this introduction, I warm welcome to Dr. Suresh Kumar. Invited him to introduce the chairpersons. Sir, please welcome, sir.
So good morning to one and all. So it is a great pleasure to introduce our resource person, Dr. R. Vadivelan, sir. Sir is completed, is a sessional professor and head of the Department of Pharmacology at JSS College of Pharmacy, JSS Academy of Higher Education and Research, Udagamandalam, Tamil Nadu. He completed his B.Pharm and M.Pharm in pharmacology from Dr. MGR Medical University, Chennai in 2000 and 2002 respectively, and earned his PhD in pharmacy from JSS University, Mysore in 2013. He is recognized as a fellow of the Indian Council of ICS, Kolkata, and having completed the FELESA accredited CCLAS course at Thanwas, Chennai. Dr. Vadivelan brings over 23 years of professional experience specializing in metabolic disorders and alternative to animal testing. He has made significant academic contributions, receiving the prestigious App-Best Teacher Award in 2019 at the APP 8th Annual Convention and 4th Indo-US Conference for his exemplary teaching and research efforts. Dr. Vadivelan has secured multiple competitive grants from reputed bodies such as ICMR, DST, SERB, and JSS AHER, organizing and convening seminars and workshops on topics including alternatives to animal testing in drug discovery and 4 hours animal ethics. He was also awarded the NAM SNT Center Research Training Fellowships for Developing Country Scientists in 2015 to 16 and was a member of the DST FIST program 2014 to 2019 aimed at strengthening sciences and technology infrastructure in universities. Throughout his career, Dr. Vadivelan has presented over 35 papers, published more than 120 national and international research articles, filed 3 international and 2 Indian patents, and authored 3 books. He has served as a resource person, delivering over 40 lectures on preclinical pharmacology across various institutions. His mentorship record includes guiding 40 postgraduate students and 7 PhD scholars, reflecting his dedication to academic excellence. He has been honored with multiple awards such as the Best Teacher Award by APP and JSS CPO and the Best Promising Coordinator Award by IPA NLB. Dr. Vadivelan is the president of the Indian Pharmaceutical Association Nilgiris local branch 18 to 24 and holds life membership with prestigious organizations including IPHA, IHPA, IPA, LASA and APP. His role as clinical investigator has led to numerous impactful preclinical studies further advancing pharmacological sciences. So now I am hand over this session to Dr. R. Vadivelan sir.
So dear delegates, சாப்பாடு வந்து 1:45 தான் ரெடி ஆகும். So another 45 minutes is there. You will try to complete before 30 minutes. You try to listen. தயவுசெய்து யாரும் வெளியே சாப்பாடு தீர்ந்துடும் தயவுசெய்து வெளியே போகாதீங்க. You try to sit another 30 to 35 minutes. Thank you.
Thank you, sir. Is it audible? Okay. Switch off light. So, respected principal, thank you once again for inviting me for this international conference. And I thank Dr. Saravanan from Suresh Kumar from SSM College of Pharmacy for introducing about me. So I think I'm not going to take a class. The first thing is I have an example for you. So what is this? This is a mobile. Put it in a silent mode or keep it aside. First thing. Okay. So listen to me first. Please keep your mobile in a silent mode or put it inside your pocket. So as your principal said, before 1:45, if you cooperate, I will complete the session. Will you cooperate? Louder. Will you cooperate? Pasikida? Illa. Thank you. Okay. Okay. We will start the session. So, preclinical perspectives in drug discovery and development. I think from yesterday's session and today morning session, you will be learnt about some of the related with the clinical practice, okay, patient related and related with the drug development process. So here my topic is very particular with the preclinical aspects. That is, before a drug enters into the clinical trials, you will be doing some of the research with the animal studies. So I cannot explain every part of it. So here specifically I will be discussing about the Schedule Y, the guideline which is followed in India. In that we will be discussing about the Appendix 3 and the Appendix 4. Okay, next slide. So what are the topics to discuss? The main thing, so the introduction, okay, about the preclinical screening in drug discovery and development, the origins of predictive animal testing. So why it is made essential to go for the animal testing. And the next, the regulatory framework. So what are the various regulatory guidelines we are following in India and in abroad? And you all know that if you are studying pharmacology in third year or in final year, or if you are a PG student, you should be knowing about CCSCA. Whenever you want to get the permission to conduct your animal studies, and that nowadays you know that we are following only the 3RS in animal testing. So in a B Pharm level, you are not using the animals. Okay, you are going with various some virtual screening methods such as call techniques and all. And my slide will go in deep with the Schedule Y, the guideline which is being followed in India for import or for the production of some pharmaceuticals which is essential for the clinical trials. And then I'll conclude with my 3 topics. Next. So you all know that whenever you use the word animal testing, it is also called as non-human clinical trial. This is useful to establish a rationale for conducting a clinical trial. You all know the 5 processes of drug development. So the first one is the drug discovery and development, clinical research. The third one is a clinical development, 4th FDA review, and 5th one is the post-marketing surveillance. So what is about the preclinical research? What do you call it as the animal testing? So the first question is asked, why we have to go for animal testing or preclinical research, and what is its significance? Yes. So whenever you are going for the animal research, it is useful for the scientific research. You will be able to know some new discovery. And if you are coming to your human anatomy and physiology also, these animal models are being used. And also to model, to study some of the disease. So you may be using some of the animals such as your rat, mouse, for studying some of the disease such as diabetes, mellitus. And also with help of that you will be developing the new medical treatment. And not only that, you will be learning about the safety and toxicity of the drugs, and also you are learning the safety of the human, animal and environment. So in this world globally, whatever you are going for the discovery, the first thing they look into the safety and then they will go for the toxicity. Ultimately, it should benefit the human as well as the animal and also the environment. Next slide. So I think you might have seen some of the images in the slide. So how many of you know the ways animals you have, we can say that what animals you know, because if I stand simply there, you guys will be enjoying with your mobile, okay? But I told you, can you say what is that animal? Any one animal can you say? அங்க என்ன தெரியுதா? தெரியுதா இல்லையா? அது என்ன அந்த ஃபர்ஸ்ட் அனிமல் என்ன சொல்ல பாப்போம்? மவுஸ். சின்னதா இருந்தா மவுஸ், பெருசா இருந்தா rat. அடுத்த அனிமல், first is mouse, second one is rat, next one is guinea pig. இல்லையா? The fourth animal is rabbit. Okay. What is the difference between mouse and rat? நான் சொல்ல பாப்போம். எந்த ஏற்படி? ஃபைனல் என்ன தெரிஞ்சிருக்கணும்? மவுஸ் சிங்குலரா ப்ளூரலா? சிங்குலர். வெரி குட். மவுஸ்க்கு பயாலஜிக்கல் நேம் தெரியுமா? தெரியாது. வெரி குட். தெரியலன்னு ஒத்துக்கலாம். மஸ்குலஸ். ஓகே. வாட் இஸ் த வெயிட் ஆஃப் த மவுஸ்? Okay, 20 to 40 gram. Good. Okay, guys. So these are the various animals which have been used for animal testing. You call them as rodents and non-rodents. Can anybody say what you mean by a rodent and a non-rodent? So we say a mouse and rat is a rodent. A rabbit or a dog is called as a non-rodent. So what is the difference between these both? Any idea? So have you seen the teeth of a mouse or the teeth of a rabbit? Okay, you will be able to see the incisor. Okay, one pair of incisor in the case of the rodents, but you will not be seeing that one pair of incisor in the case of the non-rodents. So that makes the difference. And nowadays we are using some of the alternative that is invertebrates such as zebrafish or Drosophila also have been used. And if you see the large animals which is being used in the animal research, mainly with the pigs and as well as your dog, your monkeys, and then all have been used. And for some genetic studies, you will be using some of the nude mouse and all. Okay. But for a B.Pharm student or a M.Pharm student, if you are a B.Pharm student, at least you should be knowing about what is called the small laboratory animals and what is called as the large laboratory animals. Small animals, you know, mouse, rat, guinea pig and rabbit. Large animal, at least dog, monkey should be known. And you should be able to know the difference between the rodents and the non-rodents. So once you are understanding the basic, the biochemical or the biomethodology of these animals, then only it will be very easy to understand about the Schedule Y guidelines, mainly for the animal pharmacology or the animal toxicology. Next. So always, we follow some ethics. So how are you treating the animals? Huh? How are you treating the animals? You know that if you are not treating the animals, you will be put in the jail. You know now? There are so many issues, okay, in the government. So there are so many ethics to be followed while using the animals. That's what I said. The animals should be not treated as animals. It should be treated as non-humans. Okay. You can see a picture how in center animal which is sitting with that of the human. So they should be treated as like humans only. And there are so many ethics which is being used when you are conducting the animal studies, as you may call it as animal rights, welfare, law, cognition, and the history of the moral use of the animals. Okay. So what are the ethics which is being followed in our country, and what are the ethics which have been followed in the other countries also? We will look into it. Next. Okay. So still there are some questions asked. Why are you using the animals for the research? So we are not even given the animals for normal regular academic purpose. Am I right? இல்லையா பி ஃபார்ம்ல அனிமல் கையில் கொடுக்குறாங்களா இல்லையா பார்த்துருக்கீங்களா இல்லையா இல்லை. So that is the reason. Okay, whenever you are using the animals, you should be able to give a proper justification for the substantial benefits. Why are you going to use the animals? And you should be able to know some of the issues, some specific issues, why you are using the animals for the research. And normally, they have said we are using the alternative to the animals. In some cases, if you are not having the alternatives, then only you can go for the animals, and it should be properly justified. So in the case of your M.Pharm student, when you are in a pharmacology department or when you are in another department, keep in your mind, when you are asking permission for animal studies by one committee is there in your institution, it is called as Institutional Animal Ethics Committee. So when you are asking for that, they will be asking for a proper justification. So unless you are not giving a proper justification, you cannot use the animals for the study. Next. And being a pharmacy student or a pharmacology student, you should be able to know why we are using these animals for the study. What was the tragedy made worldwide for using these animals? Okay. Previously, you know that in Europe or in USA or in the German countries, some cosmeceuticals have been produced where it has been not tested on animals, directly tested on the animals, and it has produced some of the unwanted adverse effects. If you see the first case, you call it as the Lashlier case, that is in US in the year 1930. You can see the dye which is containing phenylenediamine, when it was used, it has caused some irritation in the eye, and it has also caused some total blindness in few people. That was one of the tragedy. The second tragedy called the elixir tragedy, same in USA in the year 1937. So one company called Mastiel Company, they were using the elixir of sulfonamide. For that they were using the solvent, the ethylene glycol. So while they were using that, due to some kidney damage, it has affected more than 170 people were died. And always you will be knowing about the thalidomide disaster. I think I don't want to explain it. You know what is the adverse effect caused for the thalidomide? You call it as phocomelia. You know that the person, the child which was born with the sealed limbs. There were no limbs for that. It was a drug which was being used as a sedative, which was given to the morning sickness in the pregnant woman. You can see more than 10,000 people who have been born were born with these deformities. So after that, they have made some strict rules in bringing that mandatory. They have to go for using the animal testing before going for the humans. And then they brought another concept called as 3RS. Next slide. So the Russell and Birch, they brought the 3RS concept in the animal testing. So the first R, what you call it is a replacement, that was mostly followed in all of the academic institution and research. Okay, they brought the principles of the human endpoints. So here the word we are using is sentient and non-sentient. So what do you mean by sentient? Sentient means, so if somebody is going to prick your hand with a pin, do you are having feeling? Valikuma valikada? Okay. Same, you take a pin and you prick a vegetable. But it is having pain. We don't know. It is not able to express. That's why they are saying you go with the alternative to the animals by using some of these types of non-sentient vegetables or some related with for your experiments. So you should understand the 3 RS concept. At the end of my session, I will be coming back and I will be asking the question, at least what have you understood in my class? At least you should be able to know this. So this is called 3 RS concept in the animal testing. So the first R you call it as the replacement. So what is the first R? You call it as the replacement, where you will be able to replace the animals. The second R you call it as the reduction. What do you call? Reduction, where you are going to reduce the number of animals. And the third R you call it as the refinement, where you are going to minimize the pain and sufferings for the animals. And now as per CCCA, we are following the fourth R, that is called rehabilitation. That is, after the experiment, so this rehabilitation is mainly followed for the large animals. So whenever you are performing any animal studies or you are getting permission to do your research proposal in an animal, they will be always following this 3 Rs. Next slide please. So I was speaking about the regulatory framework which has been followed by the globally as well as in India. If you see the major guidelines which has been regulatory guidelines which has been followed mainly for the toxicology, okay, guidelines, that is OECD. Called OECD. India is also one of the member of this OECD, that is Organization for Economic Cooperation and Development. So the various acute, subacute, chronic studies that is related with your oral, dermal and inhalation, so all will be followed with this OECD. And another guideline is called the ICH, International Conference of Harmonization. Mainly they follow this QSEM, that is quality, safety and efficacy and multidisciplinary guidelines. Mainly we will be following that guidelines for the pharmaceutical product on the non-clinical testing. Okay, it has been followed from that. And another agency is called the Environmental Protection Agency, the European Medical Agency, like that various agencies are there. India will be following the Schedule Y by the CDSCO, that is the Central Drug Standard Organization, where we follow the DC Act, that is the Drugs and Cosmetics Act. So my last 2 slides I will be going with this Schedule Y guideline, mainly the safety pharmacology and the reproductive pharmacology. Next. So this is about the various framework where they followed. Okay. You know that there are some guidelines not from now, it was been followed from the year 1960, where the PC Act, Prevention of Cruelty to the Animal Act, and previously called CPCSCA. You all will be knowing it. It is called CPCSCA. Now the last year they have renamed it as CCSCA. That is the guideline what we are following in India, that is Committee for the Control and Supervision of Experiments on Animals. Okay, so that is the guideline we are following for animal research in India. Either it is an academic institution or it is from an industry. So you have to get the approval, or you call it as the license. Okay, from the CCA office to conduct any type of study on the animals. And as per the PCI, you should have an animal house where it should be licensed from this. And then 1972 Act also, you can see some Oil Profession Act and INSA guidelines. And in 1998, so you can see the gap from 1964 and in 1998, they amended a rule for breeding of and experiments on animals and control and supervision. Still we are following the rules, and these rules are modified, okay, and renewed every 5 years. Right now what we are following is the rule of 2023. So normally if you go to the CCA website, okay, it is coming under the Ministry of Animal Welfare and Department. There you will be able to see the compendium. Next. So I think I will come to the last slide, where you can see ICMR guidelines, MCI, UGC. In 2013 only they brought the guideline for that that is called discontinuation of dissection of the animal studies. And for us profession, that is the pharmacy profession, from 2015 onwards, they have mentioned the alternatives to replace animal experiments. So we all believe that nowhere in the academic institutions we are using the animal testing, but only in the case of the PG level, you will be using the animal, the modified use of animals. Okay. Next, so this is the website. Okay. Whatever the information I am giving to you, you can directly go to the website and they will be giving all the compendium, what is the rules. So always one important thing you have to keep in your mind, you should always go with the updated forms or applications. Okay. So what are the ethical principles followed by this CCCA guidelines is very important when you are going for the animal study. Point number 1. Next. So that's the history is being given. So what is the main motto of these CCCA guidelines? So I've already told you about the PC Act in 1960 and in 19. Main motto is to prevent the infliction of unnecessary pain and suffering for animals. Either you are suffering or not, but the animal should not suffer from pain and distress. That is the main motto, the main objective of the CCSA guidelines. Okay, that is the unnecessary pain or the suffering either before the experiment or during the experiment or the experiment. Either before, during and after while you are performing the experiment, not suffer from the unnecessary pain and distress. That is their motto. And I have already told you that is the rule which they brought by CCCA is breeding of vision. Okay. So finally you can say always you will be guidelines used to be updated, streamlined, and they will be giving some proper regulations for the ethical use. The main aim is to ensure humane and ethical treatment of animals. Humane and ethical treatment of animals. So there are 5 ethical principles you should be knowing. The first principle is experiment on animals. So why we should experiment on animals? The first question is asked. Am I right? The experiment you doing on animals, you know already a drug is available which is being used for treatment of analgesic. You already know it. You once again you should not be able to do it. Whenever you are performing any experiments on animals or a research, I have underlined, it is to be the purpose of the advancement by new discovery or physiological knowledge. New discovery or physiological knowledge, and also that should be able to save or prolong the human life from suffering and significant gains in well-being. So that's why they say whenever you are going to do a research on from any synthetic or any herbal anything, whether it is going to give a new advancement or it is going to prolong the life of the human or relieve from the sufferings. Next. And the principle 2 you call it the lowest phylogenetic scale. So you can see the image which is being mentioned. You know that when you are having the minimum number of animals also should be. When you are going to conduct any experiment with a zebrafish or a mouse, rat, guinea pig, they say you first initially perform with a mouse or a zebrafish. So that is the lowest phylogenetic scale in your facility if you are having. Then you will be able to go with the higher animal when it is not having. So that is called as the lower phylogenetic scale. Third principle. Next, whenever you are performing the animals, whenever you are doing a surgery or any some experiment, they said the animal should try to minimize the maybe going for appropriate sedation, analgesia and anesthesia. Isn't it? Whenever you are going to collect from animal, you simply cannot take a capillary tube and poke into the animal's eye. So let the animal be anesthetized, okay? Then you can use it. Or whenever you are going for a simple surgery on the animals, then you go for some anesthesia. Next one. Fourth one is, the most important is the moral responsibility. So whenever you are performing the research, and you should be able to know when you are going to use the euthanasia. So what do you mean by euthanasia? Any idea? So what is the difference between anesthesia and euthanasia? So anesthesia is used to produce anesthesia with the loss of sensation. Euthanasia means where you are going to sacrifice the animal. You call it as the mercy. In conditions you are going to use euthanasia? In which condition you are going to use euthanasia? See, in this condition, the humans are unlucky. You know what I mean by unlucky? The animals are lucky. Can anybody say why animals are lucky and we are not lucky? You know that when an animal is paralyzed or when there is severe recurring pain or extreme pain, okay, you understand what I am saying? When the suffering in the animal house or the animal is paralyzed or when the animals are not able to do the normal activity, they used to sacrifice. You should not use the word kill. We should use the word sacrifice. But for humans, they are not going to sacrifice any humans. Up to your death, you will be under the medical treatment. But in the animal, you don't allow that. If you know that the condition to do its normal activities, they will go for euthanizing the animals. Mercy killing. So the difference between anesthesia and euthanasia in dosing, if you say, thrice the time of the anesthesia will be the euthanasia. Thrice the time of anesthesia will be the. So can you name some anesthetic? Anybody? Any some benzodiazepines or some ketamine? Okay, you all know the dissociative. And it is not easy to get this anesthesia from the normal medical shop also. There are restrictions. Next. And so these are the method where you are going to anesthetize the by going for this method by physical method you call the cerebral dislocation or you are giving some injection method. Normally you will be going with some of the inhalation method. Next. Animals should be appropriate for their species. I think you all come for the conference, you all have been seated comfortably. I don't know why you guys are not coming front and sitting, I don't know, frankly. So always in our life we should come front, not back. Okay, so you should try to fill the front seats always. You should not get afraid. You should always, have the guts to come and sit in the front and you should be bold. Either your front or back, you and all. Okay. So for each, there should be kept in a separate housing condition. So that's what it is mentioned. The housing fit care for all. Next. So guys, these are the 5 principles. Let me summarize. One is the experimental. Okay. Why we should go for the experimental animals for advancement of the knowledge. Second is the animals on the phylogenetic scale. And third one is the proper use of animals and minimization. Fourth one is the moral responsibility. Okay, that is why you are using the euthanasia. And the fifth one is the living conditions of the animals for all. Next. See, I think I don't want to rush. Let me go with one example. So let's move. Let us see what is one method of replacement. Yes. So if you go for the replacement, so I showed the first image with a sheep and a carrot, the vegetables. That is called substitution of an insufficient material in place of a higher animals. So that is the terminology. So those are having larger brain, we are having a consciousness. But the animals that non-sentient, what we are using the greeny vegetables, they are non-conscious. Replace higher animals with the lower animals. The next slide, I'll say. So this is the first thing. The sentient animals with the non-sentient animals. So the higher consciousness. Second one. So you always replace with the higher animals with the lower animals. So mainly during your vaccine study and all, they used to collect the blood. Okay. Instead of going with the sheep, go with the rabbit. Instead of rat, you can go with the mouse. I already told you, try to go with the lower phylogenetic scale. Next. Always when you are going for the study, instead of going with handling the animal or giving the route of administration, keep in your mind, you are going with the what? It's not the real image, it is a dummy. also. So 5 years back when I was being animals, okay, by veterinary science college in Tawas, you call in Chennai, the first day they are giving only the dummy animals. You try to handle the animal, you try to inject, okay, with the dummy animals only. So always practice with the dummy only. Next. I think this is another replacement where they are using the student of the normal tissue, they will be using the chicken helium. So you know that, no? For studying the bioassay, whether you have practiced or not, guys. So you are using a software which is called Xpharma or HEB, whatever may be. We can go and use the chicken helium because we are not going to sacrifice the animal, which is already market in the chicken shop. Before sacrificing the animal, you will be using the stimulated experiment, or you may go with some techniques. So these are the possible all in vitro methods. You may use some of the cell lines. Next. The second one is reduction. What do you mean by reduction? Reduction means decreasing the number. Okay, you have to carry with 30 animals. So it's not necessary to initially go with. You can perform with the pilot studies. You can perform with the pilot. Go with 1 or 2 animals and you can check what is the observation and the result. Then you can perform the second thing. Then you can use the own controls or crossover data. You can obtain various data from the previous articles. Whenever you are going to decide, use a statistician. We have to use a statistician where he explains the power factor and he gives how many number of animals are used. It is based on your decide. What are the various parameters you are going to is important. And try to minimize the variables such as disease, diet, stress and genetics, and use appropriate species of animals. You cannot use every experiment. Okay. So for example, you want to collect the blood for 24 hours, then you can go for a rabbit, a mouse or a rat. Okay, like that it has been used. So this is the simple image I have shown. The pilot study we are going only 4 instead of 4. And always we should statistician to next. So you can go and move. So this is called a crossover study. So you have to wait for a washout period. For example, you are having one group control, another group test means. So after performing a 1 week study, you can wait for a washout period. Then once again you can cross over and study. Okay, by using the one control. And so this is the model where I show try to gather more than one experiment concurrently data. For example, you can go for the hematological, biochemical, all with one group when you are going to perform. Not necessary to go for different types of animals. Next. Okay, so always it will be very smart while you are using the strategy. Okay. For example, you might be knowing some of the tests such as student t-test or one-way and all. Okay. Next. Try to minimize the various disease, stress and genetics. So you should. Next. And try to reduce, as we have mentioned, there are various guidelines. Okay. You know that acute toxicity guidelines 420, 23, 25 have been used. Previously, we used the LD50 guidelines where we used more than 50 animals with number of groups. Now we are using only the specified fixed doses. Okay, like 5, 50, 200. Okay, 300 and 2000. Okay, next. And the final one is the refinement. First we have learned what is called as replacement. The second is called reduction, and third are called is the refinement. So while you are performing the experiments, you see that the all animals individual husbandry. Okay, and refining the experimental procedures by using some of the anesthesia, analgesics, and euthanasia, and finally it is called refining human endpoints. Next. So this is the housing and feeding for all. You cannot keep all the animals in one single cage or in one single room. So a mouse should be kept separately in a mouse room. A rabbit way of the you know, the way of keeping the rabbit was different, and a monkey also will be kept in a separate husbandry, like tree setup and all the animal house. Husbandry is different species. So this is what I was saying, using the anesthesia, analgesia and anesthetics. Next. This is one important thing whenever you use the word human endpoints. So what are you going to understand with the human endpoints? So this should be critically followed whenever you are performing your research. Okay. Wait until the animal, when you are performing the experiment. When you are performing, you know that this animal, due to more injury or pain, it may die tomorrow or day after tomorrow. So you cannot wait until the animal dies by its own. So considering that the more the animal should be euthanized. So as I mentioned, the earliest indicator, either pain or distress or a justification, and this can avoid or limit the pain and taking the actions. So that is called as human endpoint. There are separate guidelines by the OECD. Next. So I think just I'll summarize what we have discussed. So we have discussed what we mean by the drug discovery and development, what is preclinical, and why we have to go for preclinical trial. So the justification of the animal testing, and what are the three tragedies. Okay. So the elixir case, okay, the Lashlier case, and the thalidomide disaster. And then we have learned the physical principles which is adopted by the CCSA. And then we have learned the 3Rs, your replacement. Always while writing or learn the order. Okay, don't say first refinement. First thing is always the replacement, then reduction, then refinement, and fourth R you call rehabilitation. Sometimes they used to say the fifth R, I think it is learned as a responsiveness. Like that they used to say, but as per the CCSA, only third R is first, and the fourth R is rehabilitation. Somewhere one speakers they used to say fifth R is also responsibility. Okay, it is nowhere mentioned in any ethical guidelines. Okay, so now coming to this Schedule Y guidelines, I'll take only 5 minutes. Okay, there are so many slides, I'll go with only 5 minutes. In Indian system of going for your importing or manufacturing of a new drugs, we are following Schedule Y guidelines, and the main purpose is to frame guidelines for conduct of clinical research. So before conducting a clinical research, so the researcher, okay, he should submit some of the application where he has completed all the studies. Next. So these are some of the rules which are followed given by this 120A. For example, application of permission to import new drug. So like that, different code words are given for some different as per the Schedule Y. Next. Okay, coming to the main part. So whenever you are going to apply, you will be having so many items. Okay. So let me focus on the two things only. The one is called the animal pharmacology. The other one you call it as the animal toxicology. So what do you mean by pharmacology and what do you mean by toxicology? If you know only, I can go to other slides. Without knowing, then it's no. What is pharmacology? Study of drugs. Toxicology? Study of toxins. That is the harmful effect of the drugs, you call it as. Next. Okay, now coming to the animal pharmacology, if you see in the Schedule Y, it is given in the Appendix 4. Here you will be learning about the various things. In that, let me go with the safety pharmacology, general pharmacology and the essential safety pharmacology. Next. And in the toxicology studies, we will be learning about the male fertility study and the female reproductive and developmental study. Next. So, whenever you learn safety pharmacology, the answer is there itself. You are going to study the safety of the drug. Okay, you will be learning some two things. One you call it is the Tier 1 and Tier 2. Tier 1 you will be learning with the vital organs. Next. Okay. You will be able to detect the investigation potential undesirable pharmacodynamic effect of a new chemical entity, and it has also been carried prior to the human exposure. That is, you know, in clinical trials you will be learning phase 1, phase 2, phase 3, phase 4. So before doing the phase 1 trials, you will be learning. And I already told you the ICH guidelines. Okay, for the pharmaceuticals by the doing in the animal testing, that is ICH S7A. Okay, next. Okay, so I'm not going detail about it. So you, as I told you, it is called as 1 and tier 2 test. So the tier 1 is called the safety pharmacology core battery system, you call it as, where you will be learning the vital functions, okay, of mainly the central nervous system, cardiovascular system and respiratory system. You got it? So that's why it is called as tier, called the core battery system. So you know in your body which are the, all systems are important. I agree. Okay, even your digestive system is also important. You all are feeling hungry now, okay. You have to go. Every systems are important. But first system is important. What? Okay, then here means heart. What is there? Mainly you will be learning your heart rate, your blood pressure, then your respiratory system. So that's why these three systems are considered as important. Your central nervous system, your cardiovascular system and your respiratory system. Then your systems are also there, your GIT and renal system. So keep in your mind guys, okay, I'll not go about all the slides. So for example, in central nervous system, while you are doing some experiments on animals, what are the various experiments you are going to do? At least the basic, some 4 to 5. What is the follow-up studies and what are the supplementary studies? Let me go with one example. You go for CNS, you will be learning the core battery studies, motor activity. So I think you might have motor activity in your third B Pharm by using some software, which is used for motor activity. Can you say? Motor coordination. Some rod will be rotating, no? What is the name of that rod? Very good. Rotor rod? That's all. That is nothing but your tier 1 safety pharmacology study. Like that, behavioral studies, coordination, body temperature. So these are the main studies to be studied. Then the remaining you call it as the follow-up studies. Once you are completing, you will be doing the behavioral pharmacology, ligand specific, visual auditory or the electrophysiology examination. Okay guys, I think you have understood it. Similar way you will be learning for the cardiovascular system and respiratory system.
Clear.
So this is called the safety pharmacology study. So you can go to the conclusion slide. If you have any doubt in this safety pharmacology studies or in the reproductive studies, you can ask. Go back, sir. Let me say one reproductive studies. Okay. Go back. Okay. So you call it as the reproductive toxicology studies. At least let me say the guidelines and complete. Okay. Even I am also hungry. Okay. We'll go. So the OECD guideline 421. What do you mean by OECD? Organization for Economic Cooperation and Development. They follow the guideline 421. Reproduction, development, toxicity and screening test 422. So they have combined the both the male fertility study with the reproductive developmental toxicity screening test. So generally you call them as DART studies, D-A-R-T. Next slide. So this was followed by FDA guidelines in the 1996. And whenever you are learning the reproductive toxicity studies, guys, remember they call it as the segmental approach. Segmental approach. At least you know what is segment 1, segment 2, segment 3. So the segment 1 is called the fertility and the general reproductive performance test. Segment 2 is called the teratogenesis test because it is mandatory to study the teratogenesis test because I have told you the tragedy of the thalidomide disaster, and segment 3 is the peri or the postnatal segment. So for all these toxicology studies, you will be having the updated guidelines from the OECD. Okay, you can, there is a website for OECD. There is a separate library also. So all the guidelines are given there. Next. So this is the principle behind that. You can move. You can go with that chart. So this is the chart which is being given where you will be able to know about the male reproductive studies, female reproductive studies, and then you will be able to know, that is what I said, segmental approach, 1, 2, 3 guidelines. So normally you can see the animal will be given mating, then after pregnant, so at least the female will be given treatment for some 63 days. Then after lactation also, you will be checking the endocrine disruptors, thyroid hormones and all will be checked. So this you can follow it. Final slide next. Okay. Now I am coming to the conclusion of the slide. Okay. So now in today's world, let me say what I have completed. So I have just made one ethics in animal use. We followed the CCCA guidelines and what is 3RS. Got it? Your question rounds come. And I was making a little brief introduction about what is Schedule Y and what is safety pharmacology and reproductive pharmacology. Next. So in today's always, you know, all people always, wherever they go, they are speaking about what? What are you doing with your mobile? If I ask a question to you, what will you go? Where will you go? Huh? You will just either open the ChatGPT or whatever Gemini. In different mobiles, some different AI technologies be given. So you all are mostly interested in technology only. Even the PCI syllabus, they are trying to bring in every semester one AI. I am not an AI specialist. Okay. But I can say something about AI related with the pharmacology. First thing you all should know the algorithm. Python language. Because we are learning that language itself is not easy. You are a pharmacy person. I don't know how we are going to incorporate it and you are going to learn. But no other go. You have to learn. In case of pharmacology, for example, here I am there to see the behavioral status of the animal. I can able to see you are talking, he is talking, the animal is moving. While you are using the AI technology, what happened? I will not be knowing. The HI. HI means human intelligence will not be looking to it. Only AI will be looking. They will be giving the report. Okay. So that case is sophisticated. You have to learn the algorithm. Takes time to understand. But definitely the future will go with the AI. Okay. So thank you guys. I think I have tried to complete the session on the time. If you have any questions, you can please ask. And I won't ask you questions because the only question is, we all are hungry. We have to feed ourselves. Okay. Thank you.
So thank you very much for your session, sir. It might be our students are understand about the ethicals. So sir explained very clearly about the OECD guidelines, CCSA guidelines, ICH guidelines, and also Schedule Y, CDSUO guidelines. And also sir very explained about the 3RS and 4RS. So it will be useful for your future research projects. I thank you very much, sir, for this wonderful session. Thank you.
Of our respect and admiration, we now take this opportunity to present a memento to Dr. R. Valen, Professor and Head Department of Pharmacology, JSS College of Pharmacy. I invite our chairperson, Dr. P. Suresh Kumar, sir, Principal, Sanmugha College of Pharmacy, those person. I would like to invite our principal, Dr. R. Sambat Kumar, to honor our chairperson. I invite Mrs. Vanita to present a memento to our chairperson. இதெல்லாம் ஒரு சான்ஸ் கிடைக்கவே கிடைக்காது. நாங்க கொடுக்கறோம் பாருங்க. ரெண்டு பேரும் மேட்ச் எல்லாம் ட்ரெஸ் பண்ணிட்டு வந்திருக்கீங்க. டேங்கப்பா. நம்ம ஸ்டூடெண்ட் இல்ல சார், அதனால தெரியாது. Thank you, sir.
ஸோ சார் வந்து டாக்டர் சுரேஷ் அவங்க மிஸ்ஸஸ் வனிதா அசோசியேட் ப்ரொஃபஸர் ஈரோடு காலேஜ் ஆஃப் ஃபார்மசி. அதனால தான் அவ்வளோ வெக்கப்படுறாங்க.
Now break for lunch. We request all the delegates and participants to kindly designate. We request you to reassemble promptly by next session. Thank you.